Hello, my name is Sherry Dang, and my presentation is on a feasibility study for using large amounts of single-cell data to monitor biodiversity. The rapidly evolving field of bioinformatics provides innovative tools and methodologies to address some of the most pressing environmental challenges of our time. One such challenge is the effective monitoring and understanding of biodiversity within various ecosystems. Biodiversity is crucial for maintaining ecological balance, yet it is increasingly threatened by human activities and climate change. To protect biodiversity, it is essential to develop robust methods for analyzing and interpreting the vast amounts of biological data generated from diverse species. This project on classifying cell types for single cells in humans serves as a pilot study with significant implications for environmental bioinformatics. This research focuses on the detailed classification of cell types, leveraging advanced computational techniques to handle large datasets of over 5 million cells. The methodologies developed and refined through this study are not only applicable to human cells, but can be extended to a wide array of organisms, thereby enhancing our ability to monitor and preserve biodiversity. One of the primary challenges in bioinformatics is the management and analysis of large datasets. Single-cell RNA sequencing is a powerful technology that measures the expression profile for individual cells in large quantities. Clusters of cells can be obtained using single-cell RNA sequencing, but manually annotating cell types is difficult and time-consuming. Effective classification of cell types from such data is critical for understanding the complex cellular diversity within organisms. This capability is particularly important for environmental studies, when sequencing of multiple species can provide comprehensive insights into ecosystem health. This study demonstrates the feasibility and effectiveness of using advanced bioinformatics tools to classify cell types from large datasets. By establishing a robust framework for data handling and analysis, the groundwork is laid for future application in environmental monitoring. The techniques and insights gained from this study will be invaluable for scaling up to more complex and diverse datasets, encompassing a wide range of species and contexts. First, the data will be downloaded from databases and converted into expression matrices, with rows as the genes and columns as the cells. Then the data will be split by dataset to prevent the same cells to appear in both the training and test set from the same dataset. Then the model will be trained and evaluated using deep learning methods. Since the performance of the model can be affected by ambiguous annotations in the data, propagating all annotations of a child to its parent cell will improve the accuracy of the model. To implement hierarchical correction, we'll create a table of parent and children cell types. This creates an annotation tree for each cell so it can be annotated to its parent cell types. The depth of the network can also change the accuracy of the model, so experimenting with different network depths can improve the model. Building on the success of this pilot study, future work will focus on several key areas. First, expansion to diverse species. Extending the single-cell classification methodology to a variety of species across different ecosystems will involve adapting the computational tools to handle species-specific data and characteristics. Similarly, this model can be expanded to disease cells, which can impact developments in health sciences. Also, development of bioinformatics tools will also improve this model. We can create user-friendly bioinformatic tools and platforms that can be easily accessed and utilized by researchers and conservationists worldwide. These tools will facilitate the analysis and interpretation of large-scale biodiversity data. This can also be applied to conservation efforts. We can apply the insights gained from single-cell analysis to inform conservation strategies and actions. This could include identifying key species for conservation, understanding the impacts of environmental changes on cellular functions, and developing targeted interventions to support ecosystem resilience. Thank you for listening.